Okay, so welcome to a new episode of Future Foods Interviews. On this episode, we are speaking to two esteemed doctors. One, Estere Sienkmane, who is co-director and program manager for Cell Ag UK, which is a non-profit organization. Looking at, I'll ask Esther to introduce a little bit more about that in a second. And she's also a cell computational biologist. And also joining us is Dr. Alice Esperanza, who is a senior tissue engineer at Cell Agriculture Ltd. So you may notice that the names are quite similar, but they are working in similar fields. So it's perfectly acceptable in this situation. So, first of all, could we get an introduction from you both, a little bit as to how you got involved in this area of cellular agriculture, and what you do really at your kind of separate workplaces. So, Esther, would you like to go first?
Sure. My personal background is kind of academic in cell molecular biology, but my involvement in Cell Ag really started in early 2022 when I joined Cell Ag UK, which is a small non-profit. So it's more to do with community buildings. So we work closely with academics and professionals and students to try to kind of connect people with each other, create resources for them, like just a map of what is happening in the UK. And we run programs like mentorship and student conferences and outreach activities. So we've kind of expanded since that over these last 3 years. So it's been exciting to do more and see this field grow in the UK as well.
Okay, thank you, Esther. And Alison, just a brief introduction to you and your organization.
Yes, so I'm part of Cellular Agriculture Ltd, which is a company, and we develop bioreactors specifically for cellular agriculture, although we are branching into other things as well. And as a tissue engineer, my day-to-day is a lot of experiment planning, but then also working with the reactors themselves, doing optimization stuff with the cells and sometimes with the media. So it really spans a lot of different areas of scientific interest, which is why I like it so much.
Okay, so and Alison, who did you work for before? I seem to remember you have quite a history in this area, right?
Yes. So yeah, I first heard about cultured meat back in like 2016 when Memphis Meats, now Upside Foods, put out their first cultured meatball. And ever since then, I was like obsessed with the field. So I started cold emailing people in 2018 and I ended up going to a conference and that's where I met Marianne Ellis. And I ended up going to be a PhD student in her lab because I wanted to study cultured meat. And so I did my PhD with her and now I work at her company.
Okay. And I know Esther, because we have collaborated at the festival, the Eden Festival in Scotland. We're on a panel there discussing cultivated meats, which was fun. And also we both met Alison at the Science Museum in London for the Fair breakfast meeting. Fair is part of the sort of collar investment scheme, which was also very interesting. Now, I want the reason I've brought you both together is because we're going to have a bit of a lesson, I suppose, for those of us who are not scientists. We're going to we're going to understand how it all works. At least that's the objective. So I've brought these two wonderful and very knowledgeable people together to explain how it happens and what happens. So let's start. Esther, maybe you can start us off. Uh, what exactly is cultivated meat, and how does it differ from plant-based meat alternatives?
Yeah, so I guess your podcast listeners would, probably know the answer to this question. But in any case, so meat, it's like, as we know it, it's a piece of animal tissue, right? But you take it from a live animal. Whereas the idea with cultivated meat is that instead of taking a chunk of live animal, you take some cells and you grow them into, those tissues. So it could be muscle tissue, it could be fat tissue, it could be some connective tissue, it could be a mix of everything. But whichever you think is the most relevant for your product, and then you can grow these cells outside the body in bioreactors in process called cell culture or tissue culture, and then form it into a sort of, whichever product, a mince, a meatball, a steak, a burger. So there's various methods for doing the various steps of these process and various cell types, but essentially that is what the idea is.
Okay, so it starts from a cell, right?
Yeah.
And where do you get that cell from, and how do you take that cell? That's open to either of you, I suppose, or both of you, in fact. So yeah. So what are the starter cells, and which are the best types, maybe for?
Yeah. I mean, I guess maybe continue answering in general, and then Alison can share which kind of cells you work with, if you can share. It's a Cellular Agriculture Limited. But so as I mentioned, there's I guess, the three main tissue types that people generally work on. It's muscle cells and fat cells and connective tissue cells, which are called fibroblasts of. So you can take what we call primary cells. So these are cells taken directly from an animal. And in our bodies and in animal bodies, we have kind of cells at different stages of what we call differentiation, so different levels of maturity, like let's say neuron or skin cell is like a differentiated cell. So it's cells that already decided what it's going to be. Like I'm going to be a skin cell, I'm going to be a brain cell, a neuron. But there are also cells that are kind of half decided what they're going to be, which are called adult stem cells. So it's a cell that can differentiate either into anything. So there's something called pluripotent stem cells, so cells that can differentiate into any type of cell. And that's how, embryos grow and form different tissues. Or it could be kind of a half specialized stem cell, which is like, I can become a muscle or fat, but not really anything else. So then because one of the properties of stem cells is the divide, which is very useful when you're trying to grow cells for an application such as cultivated meat. So it's usually these type of stem cells that are used for cultivated meat productions, and it depends. Various companies use various stem cells, and then obviously it also depends which species you're using, because you can take cow cells, and then you have to find a place to take it from a cow muscle or from somewhere, or you can use like chick cells. I know that like for chicken, you can take cells from an egg, for example, because again, it's a biological material, it has cells. So yeah, there's a variety of different things you can use. I don't know if Alison, which type of cells have you worked with?
Yeah, I was going to say this is a topic of great interest, and I would say a lot of debate in our field is whether you should be using primary cells, so where you take a biopsy and you just get some cells out, you use those, or if you're going to be using what they call immortalized cells or genetically engineered cells. So, for example, one of the cell lines we use in our lab has been around since like the 70s. It's an immortalized mouse muscle cell line. And that's like our workhorse where we test a lot of things with that. And then maybe we try it with the primary cells, which are more sensitive and maybe a little more difficult to work with. And so, yeah, it's there's a lot of different options, and it depends a lot on the regulatory concerns, I think, because obviously, if you genetically engineer your cells, they can grow really fast and they can be more nutritious, all of this stuff. But then, of course, it poses its own challenges. So, yeah, there's a lot of give and take with what cells you use at the start.
Okay, so I mean, there's a lot of questions uh, to follow up there. I mean, so in regards to kind of genetically engineered, what's the difference between that and genetically modified? Is there a
Oh, yes, that is a good question. Do you care to answer that, Esther? Or I could give it a crack.
Yeah, I think it's part of it is science and part of it is kind of publicity, I think, because obviously there's, people talking about genetic GMOs and genetically modified organisms, and that was the first kind of technology that was introduced, and then it was kind of to some extent mishandled both by kind of corporate interests and also by public. There's been a lot of misconceptions about what genetic modification is. And then, when you ask people about like, oh, would you eat a banana with DNA in it? They were like, oh no, I would never do that. But all plants have DNA in them anyway. So as far as I know, the kind of the newer technologies are often come under the label of gene edited, which
Mhm.
means that it's a more precise, now that we have technology such as CRISPR and these other what's called gene editing technology, where we can change things in a more precise manner. So it also matters whether you change a gene or whether you introduce a new gene. So there's all sorts of technicalities around this, but And it's also like the traditional breeding of plants. It's, you know, it's also changing plants' genetic material. You're just doing it by breeding and by inducing mutations, like by UV, but yet that doesn't count as genetic modification. But if you induce mutations by another means, it does count as genetic modification. So it's really, it is not a very clear line, but these labels have, yeah, have some weight in terms of obviously public perception as well.
Okay. So the kind of public perception of GMO, you'd say, is largely kind of misunderstood, would you?
Yes, unfortunately, and there's a lot of I think golden rice is one of the kind of good and sad examples. So it's rice that has been engineered to have vitamin, I think it's vitamin A, and it can really help in malnutrition, especially in the more developing countries. But yet a lot of the like organizations and even like environmental organizations are against it and say that, oh no, it's bad, even though it has some clear health benefits. But it's I think you can do a whole other episode about genetic
Right.
engineering and
Yeah, no, we
modification. So
have to be
maybe we
careful
shouldn't go
not
too deep into that.
to be careful not to go too far because we're not talking about crops, are we? We're
Yeah.
talking about
Yeah, but I
animal
guess
cells. Yeah.
I guess what Alison was saying, like there's technologies to help because this coming back to this issue between stem cells and primary cells, like primary cells are harder to grow and they're not designed to proliferate to grow because when like adult tissues start to proliferate in human bodies, that often becomes cancer. Like that's like they have primary cells have mechanisms to not do that. So, and like, how do we, yeah, keep the primary cells, but also try to grow them? Like, that's the question. And that's where some of these genetic technologies can help us in a very controlled way to like control some of the genes to help us, to help the cells grow, but also for them to do it in a controlled way. But
Okay.
then,
Okay. I mean, if I can say how I understand it is that cells are taken from a fetus generally, or at least historically, right? The cells that people have started with have been taken from a fetus. Is that right? And if so, why? No.
That was definitely the most common a while ago. Like young cells are a lot easier to convince to do things. Um, but I think now it's fairly common to just take a biopsy from an adult animal or my PhD work actually I took meat out of supermarket shelves and I just got the cells out of that. Um, yeah,
Okay.
so there's definitely other options. Or there's a very famous example, I think it was Just Foods. They do some culture of meat stuff. Actually, they had the first product ever onto the market, which is in Singapore. It's like a chicken nugget. But they did this.
It's a chicken feather.
Yes, it was the feather. So they took
Yeah.
they took cells out of a chicken feather. So it didn't even need any sort of like surgical intervention. And then they just reprogrammed them, which is something you can do genetically, into muscle cells. So they could grow muscle from something that a chicken had naturally just shed. And the chicken was there at the chicken nugget party, which I always think is so cute. Those pictures are
Yeah.
really cool.
Because that
that
was
is
going to
very
be
cool.
my next question, actually, to ask. So clearly, with that feather, that was edited. That was gene edited, right?
Yes, I believe so. It was reprogrammed, and it would be very, I mean, I think it's technically possible to reprogram without gene editing, but it's very difficult. So I would assume they did some sort of gene editing. But I don't think they said specifically.
Yeah, and there's a lot of depending who you ask, because I've asked scientists on this program whether that's possible, and they said it wasn't. But you but you say it was.
Reprogramming cells? I think
Well,
it,
for the
yeah.
chicken feather, to kind of take a feather and from that grow meat.
Yes, I think it's possible.
Okay, okay.
Yeah. Do you agree, Esther?
I, I think it really depends on what, I mean, I'm not that familiar with specifically like what kind of cells you find in chicken feather again. Like it's very specific. And also there's also some interesting technology, for example, like I know this is just one example, like a company Meatable in the Netherlands. they have this something called Optiox technology, which is another type of reprogramming cells. I'm not quite sure what is involved, but I know
Yeah.
it's like a patented technology that's being used in stem cell, like in cell therapy, and they've also like in Cambridge. So I've actually came across them through like other collaborators I've had in Cambridge, but they license the same technology to Meatable to create cultivated meat. So there's all sorts of these technologies to allow you to do things in more controlled way with or without genetic engineering. So it's a quickly developing field, and there's a lot of interest um, in that. For example, there's a whole network called Accelera. If anyone's interested in specifically cell line engineering, like there's a network for cell line engineering, cellular agriculture, actually also run by people in the Netherlands. So yeah,
Okay.
that's like people share papers and discuss the newest kind of cell line developments.
Okay. Well, one more question on cells, and then we'll move on. Just a quick idea. So, the way that cell technology, I suppose, is growing. Where do you think it's going to be in sort of 5 or 10 years from now in regard to sort of cellular agriculture? How easy will it be to sort of take cells and grow them? Or how
Well,
much?
it's certainly getting easier all the time.
Yeah.
And the wonderful thing that our field benefits from a lot is that we get to learn from medical research. And a lot of that gets published in really prestigious journals. So you get amazing information. And so as cell therapies, which I think that's what Esther was
Yeah.
just referring to as well, as cell therapies become more widely researched and more common, then those technologies are things that we can sort of adopt into our own industry. We just have to find ways to make them cheaper.
Yeah, I think it's a like, good question about the cost and the technology because, like, there's lots of technologies that exist. And the thing is
Yeah.
that for cell ag, you need something that's also cheap. And then there's interaction between the type of cells that you use and the type of media that those cells need. Because, again, like stem cells, for example, often need a lot of growth factors, and the growth factors are very expensive. So, like, you either have to make cheaper growth factors or you have to make cells that don't rely on growth factors so much. So there's like always this interaction between other components of what we need to do, right?
Okay. Okay, well that brings us quite neatly onto the next topic of growth media. So, what is a growth media, and and is it necessary for the growth media to be the bovine serum style, or can there be alternatives? Who would like to take that one?
I'm happy to, unless you want to.
Okay, well, let's have a bit from yeah. If you'd like to start, Alison, and Esther,
Yeah,
if you have
yeah,
something to
sure.
add, yeah.
Yes. So I would say that the growth media is what provides the cells the environment they like to live in. So yes, it's a lot about nutrients and what you're feeding them, but it's also about how salty it is. It's about the temperature. It's about the pH. So the cell culture media maintains all of those things in the perfect conditions for the cells. And it's full of things like amino acids and nitrogen sources like glutamine so they can build their proteins and their nucleotides, and then sugars so that they can get their energy and get their carbon source. And one of the things that's in there in as standard is, like you said, the bovine serum. But it's not necessary. It's largely a cocktail of growth factors, proteins, but it's very ill-defined. Um, Making chemically defined media, or even just animal-free media in general, has been something that the medical fields have been pursuing for a while. And it's definitely possible. It's expensive usually because you're getting these purified recombinant proteins instead of this mixture that just comes from slaughterhouses. But it's definitely possible, and a lot of people are developing serum-free media.
Okay.
Yeah, I can continue with that. Yeah, so on the like, I guess just a slight explainer for it. So recombinant is when we produce a protein in a biological system, so often in microbes. So, like, let's say insulin for type 2 diabetes, for type 1 and sometimes type 2 diabetics. Um, in the olden days, used to be taken from like pigs' pancreases. So, like, you would mash up insulin is generally in animals produced by the pancreas. So, in the old,
Yeah.
like before 70s, like they would just mash up pancreases from pigs and purify insulin because that was the only way to treat, diabetes. But since we have this recombinant protein technology, we can take the insulin gene, put it in a bacteria, E. coli, in a yeast cell, and then that that bacteria or that yeast would produce insulin for us. And it's actually much cheaper now than, growing the whole pigs. And that's coming actually to the precision fermentation side of things, which is another sub area of, cellular agriculture. So the problem with media for mammalian cells is that it's just like a mix of many, many things, and we don't even know often what exactly is the role of specific things. So, it's not a single protein like insulin. Like we can produce the individual components. It's just defining what exactly those components, like which ones we need desperately, like some growth factors, and which one maybe we can do without, or we can find other substitutes. So, for example, there's a lot of work on like plant hydrolysis or
Yeah.
other like yeast hydrolysis of like taking other biological material, hydrolyzing it, basically like breaking it down into components, because that should have amino acids, that should have some useful stuff for mammalian cells to take. So there's a
Okay.
lot of work on that, and as far as I know, most companies are moving to FBS free, animal component free media.
Yeah. But there's a lot of secrecy around it, I suppose, because it's almost it's the holy grail for lots of companies, so they try and keep it secret.
I think there's also specific companies that develop cell media that just focused on developing cell media. There's one in London, for example, called Maltus. There's other ones.
Yeah.
I think it's a bit of a also different again. Different cells need different media, so it's not like a single problem that will be solved once and then it's solved. It's different depending on exactly what you're doing. You'll need slightly different things. So it will be it's something that people are very aware of and working on, but it will be an ongoing thing.
Okay.
Yeah. And just to shout out the nonprofits in our space for exactly that reason, it's great when we get publicly funded research into this sort of stuff because it means people don't have to keep reinventing the wheel. And so there are already published papers that have been funded. For example, I was funded as a New Harvest fellow. So I was funded by New Harvest, which is one of our big nonprofits. And one of their other fellows, he's published Beefy 9, which is a serum-free media formulation, and then an improved version, Beefy R. And they, in turn, are built off the backs of Essential 8, which is a very common iPSC serum-free media. So there is a lot out there publicly in the literature, and sometimes companies will even publish their research. I'm pretty sure Mosa has published a couple of papers on serum-free media as well.
Well, apparently most of them have to because they're part of a sort of academic institution, aren't they? So it's kind of that's
Yes, I
why.
think they get some funding, and so
Yeah.
the students that work there publish something like that.
Yeah. So what is the bovine serum then? Where do you get that from?
Yes, it depends on the type of bovine serum. So if you use fetal bovine serum, that comes from calves, basically pregnant cows are discovered at slaughter, and then you do cardiac puncture or some other methods on the unborn fetuses that are discovered, and that's where the blood comes from. But some people use adult bovine serum, which is just from donor animals that are adults.
So serum is like a protein portion of the blood. So,
Yeah.
in the blood, there's blood cells like erythrocytes, white blood cells, red blood cells, and then there's like proteins and nutrients that are just floating in the blood. But again, like FPS is just something that was so fetal bovine serum is like extremely common in biomedical research, but it's more for historic reasons that it was like an easy source to just get this concoction that is used for cells to grow. But it, like as Alice mentioned, like it's actually people in biomedical research don't like it that much either because it's very variable, right? It comes from a biological source where you can't control what it actually is, and the concentration will always be slightly different of various components. So if you're trying to make a defined process for something, it's actually much better to have purified animal-free components. And growth factors is one of the main components. Well, it's one of the main components that people are interested in within serum, but it's actually tiny, tiny proportion. You only need like tiny, tiny amounts of growth factors because they're signaling molecule. It's not something that cells use as a nutrient. It's something that cell use as a signal to tell them, okay, I need to grow like this.
Ah,
And then the other stuff in serum, yes, there's other proteins such as albumin and transferrin that have other functions and could possibly be replaced by other sources, or again, could be produced recombinantly.
I see. Okay. So we know what the cells are. We know what the media is. So when you take your cell sample, what do you do with that to make it grow? And then when it's got to us, what stage in its growth do you then put it in the media, or is that, how
So
does that work? Who would like to?
maybe I can start, and then Alison can
Yeah.
tell about the more
Yeah.
expansion
Yeah.
stage. Just because
Okay.
when we were at the Science Museum, when we all met at this opening of the exhibition, which I would recommend to anyone, there was this stack of flasks that these sort of plastic flasks with pink liquid, and this whole thing started with Alex asking me like, "Oh, what's that?"
What are
Because
they? Yeah.
this is like an extremely common sight. You would find them in any cell culture laboratory, but it's completely non-obvious to anyone who hasn't been to a cell culture lab of what that is. So basically, as soon as you take cells out of the body, like they need nutrients, they need, as Alison was saying, the right pH, the right salt concentration. So you have to put them in something immediately. And cells also like to attach to something, unless they're already like floating cells, like blood cells that, floating in your blood. Cells like muscle need to be attached to something. So, and actually, conveniently, perhaps in the lab, most cells are fine just attaching to plastic. So the way we grow cells in the lab is, we could take a biopsy or something and then kind of split a bit that suspension so that cells are a bit separated a bit between each other, and then we can put it in media. So put it in this liquid that's often pink because of the pH indicator in it. Put it in this pink liquid with some nutrients and put it into a plastic flask, and they will just attach to the bottom of the flask. And they will just start growing and dividing if they're in the right state. If you provide the right nutrients, you have to change the media because A, they deplete the nutrients, and B, they also release the byproducts if they reach a certain concentration, that also can be toxic for the cells themselves. So you need to change that media, provide new things, and then you can expand it to higher and higher surface area if you're growing them on plastic. But then for cultivated meat purposes, at some point you need to move from growing them in this sort of attached manner to growing them in bioreactors, which is a suspension, like a big liquid. So I guess Alison can tell us more about bioreactor stuff.
Yeah.
Yeah, so once you've got enough cells that can go into a bioreactor, and there's bioreactors of all sorts of sizes. So usually you would start with something we would call a bench scale bioreactor, which is just a bioreactor that will fit on the bench in the laboratory. But then you get bigger and bigger and bigger. So some of these flasks that you see, the largest ones people usually use are called cell factories, and they're just layers like a hotel where the cells are living. And we still call that two-dimensional culture. So anything in flasks, we call it two-dimensional because they're just sitting on a flat surface and that's all that there is to it. But when you move into three-dimensional culture, they're also occupying the Z axis, I guess you could say.
Okay.
So you can do that by moving them into reactors where they're floating in suspension, as Esther said. But if they're adherent cells and they need to attach to something, they'll have to be sitting on microcarriers. So those can be made of all sorts of different materials, maybe the same plastic that was used in the flasks. Or the type of reactor that we use is called a hollow fiber bioreactor. So it's essentially full of a bunch of like drinking straws, you could think of them that way, that basically recapitulate the in vivo environment where you're thinking about capillaries and veins and arteries. They serve that same function. The cells are growing on the outside of the straw, and we're passing the media through the inside of the straw to feed them directly, just like your blood is carried throughout your body. And that way, using these big tanks full of microcarriers with cells on, or these huge, well, not even that huge. That's the nice thing about ours is they give you really high densities in a small reactor volume. But these different reactor types can give you much, much higher surface areas, so you can grow much, much larger numbers of cells.
Okay. So those straw-like things, are they what you would call a scaffold, or is this
Yeah,
different?
yeah. I think anything, I would call anything that the cell will attach to and grow on, that would be, I would refer to that as like its scaffold. So
Okay.
tissue culture plastic is a scaffold as well.
Yeah,
So
but I
there's,
guess when
yeah, go
people
on.
talk more generally about scaffolds, like I guess in cultivated meat application, as far as I understand, it's also like or tissue engineering application is often that could stay in the final product as well. So if you have a plant-based scaffold or a mycelium-based scaffold, then that could also stay. Like just as in your steak, you have some kind of connective tissue or even some bone which the cells are attached to. You can have the same for your cultivated meat product where you have something else that the cells are attaching to, but that also forms the final product.
Yeah.
Yes, I love the idea of having edible scaffolds because getting the cells off of the scaffolds if they're not edible isn't always the easiest thing.
Well, that's what I was going to ask. So, how do you do that? So, if you could just sort of tell us, Alison, quickly, what products are you making there at Cellular Ag?
Yeah, well,
Uh,
our company is, we're not like a full stack company, so mostly we just develop the bioreactors and we
oh
want to
yes.
be a place that people can bring their cells and their products to sort of try in our reactors and see how we can, do the engineering part and help with that. So we're more on that end. We don't really make our own like
Okay.
meat
So,
products.
but companies that make cellular agriculture products, meat products, are they generally going to want to have an edible scaffold as part of the product? Is that a requirement, or I mean, are you offering a different sort of product, if you know what I mean?
Yeah, I would say I don't think it's a requirement that the scaffold be edible, but it has its own, yeah, so it will end up with its own challenges, but it also has its benefits. I'm sure there's been a lot written on this, but basically, like if you have an edible scaffold, that's great, but obviously a lot of your product then isn't really, it's not the meat. Like you might have, 90% of the mass that you end up with is your scaffold and only 10% is the cells. And
Mm,
personally, I think that's totally fine and still really cool. But if you wanted a pure meat product, then unless the edible scaffold is degraded, which is another option, then you would have to find a way to get the cells off and have the cells be purely that, that's your muscle stuff. Yeah. But
okay.
there's all sorts of different options. You can have your edible scaffolds, you could have reusable scaffolds where you're getting the cells off and just the scaffold is left behind, or you could have a scaffold where you can dissolve it and it will just go away. Um, yeah, there's all sorts of different stuff.
So I mean, yeah, so the bioreactors you're working with, they are kind of fiber uh, based, kind of optical fiber based. So how does that differ from the bioreactors other companies are using?
Yeah, I don't know everything about what other companies are using, of course, but it's my impression that it's much more common to either adapt your cells to suspension so they can go in the conventional suspension reactors, or to put them on microcarriers, which are little spheres, and then stir those in suspension. I think those are the very common ways to do it because suspension cell culture is well established. It's been used to make antibodies and enzymes and stuff for decades. So, adapting meat to work in those systems is what a lot of people have gone for, whereas we're trying to like sort of make a new system built for meat, kind of thing.
Yeah, I
Okay.
guess what Alison is saying as well is like bioreactors is not some like it's not a new concept, right? It's existed for biomedical application. It exists for food applications, right? Like yeast and beer.
Yeah,
These things
yeah,
are grown in bioreact like the metal things you sometimes see in breweries and in like hipster bars that have like their own microbreweries. Like that's a bioreactor, and that's often how at least from like kind of the pictures I've seen from like companies, like that's often how it looks in cell cultivated companies as well. It's just adapting mammalian cells to grow in that sort of stirred tank, what they're called. So because
Hmm.
it has to, you kind of have to stir it so cells don't stick to each other because otherwise they will just also clump together
yeah.
and there
And they're
won't be
dense,
enough
so
also
they sink.
diffusion of nutrients. So I
Yeah,
think that type of reactor is called stirred tank bioreactor, and it's just another type that's quite commonly used, and there are its own challenges and its own opportunities.
Okay. And in those suspension bioreactors, then they would need to have a scaffold because otherwise they'd stick to the sides or something, right?
Well, I
yeah,
guess
it
Alison
depends.
mentioned the microcarriers, which is the one of the other main. So it's microcarriers,
Yeah.
I guess you can say it's a type of scaffold, but it's these like
Okay.
Yes.
small spheres that, are in suspension themselves as well. Or you can also try to convince the cells to grow without anything, which is, again, like other cells do it. Blood cells don't attach to anything and just circulate in the blood. So in
Yeah.
theory, other cells might be able to do it. It's just muscle cells don't normally do it.
Okay.
And the really cool thing about muscle cells, which you can observe so easily under a bright field microscope, and I recommend anyone look up pictures or videos of this. But when they start differentiating, which is what Esther was talking about before, where they're maturing into a very specific type of cell, they fuse together. So they initially are single cells, and then they'll fuse into these long myofibers, and then those will collect oh sorry, they'll fuse into myotubes and those collect into myofibers. And that sort of structural conformation is difficult to achieve if they're in suspension because that, as far as I know, does require a scaffold. Cells in suspension can't do that type of differentiation.
Okay, so what is the difference then between I suppose not what's the difference because there's many differences, but what's which are the easiest cells to grow, like fat, muscle, or fiber, I suppose. I mean,
I would
obviously,
probably say fibroblasts. Have you
okay,
worked with fibroblasts, Esther?
Yeah, I've worked with fibroblasts in the lab. They're like much less fussy. Like it's so fibroblasts form connective tissue,
Yeah.
and they're just like a more easily accessible type of cells as well, and more just they usually don't require quite as much, and
Hmm.
they like to attach to things because, but then they are so then I don't. There's probably a lot of challenges growing them in suspension, but they're in terms of their media requirements, they're simpler. Whereas fat cells might be more adapted, might be possibly easier to grow. I would want to be careful about that claim, but possibly easier to grow in suspension. But they have specific media requirements, but they're simpler in a way because they collect the fat in these fat droplets, and as long as you've got to provide the lipids, that they need to make those fat droplets, it's sort of fine. They don't do that much else. Whereas muscle cells, as Alison was describing, have these more complicated differentiation steps.
okay. And there's a bunch of companies now that are putting that are creating cultivated fats.
Hmm.
Now I've heard from someone that that's the easiest one to grow, but you're saying not necessarily. So why are these companies creating fats more than, rather than meats?
Well, I think the thing about fibroblasts being easy to grow is, or relatively easy to grow, is that they're not necessarily what you would think of as meat. So I think it depends on what you're trying to achieve, like from, are you trying to exactly replicate what conventional meat is? In which case, fibroblasts alone can't really do that. And you would need muscle, you would need fat as well. Um, I've personally never worked with fat cells, so I can't comment on if they're easier or harder than muscle, but muscle definitely has its challenges. But of course, meat is mostly muscle cells. So if we want to make meat, got to figure it out.
I think,
Okay.
as far as I understand the approach that these sort of cultivated fat companies are taking, it's like it's a specialized type of cells, which, some things are easier, some things might not be as easy, but it's known that it's very important for the flavor. So even
Yeah.
if you can't replicate the whole meat by just growing fat, or even fat plus fibroblasts would still not give you meat because it would lack the muscle, you can replicate a lot of the flavor and
Yeah.
possibly even do some nutritionally interesting things like adding healthier fats, that even healthier fats than would have been found in a conventional meat, and then combine this with plant-based product so that would be like a tastier, sizzling, healthier bacon, for example, that just has the cultivated fat rather than having the whole cultivated bacon. So I think that's the approach that a lot of the cultivated fat companies are taking.
But as I
Yeah.
understand it as well, a fat does not, it can be a liquid form because I mean, when your product is like is like a soup in a way, in the bioreactor. If you're stirring it, it's like a biological soup. And I suppose the fats that you create don't have to look like meat because
Yeah,
they're
I
added.
think it's
Yeah.
also there's also a difference between like adipocytes, which are the fat cells, like in your body, you have the insulation layer that, it is cells that have these droplets of, fat. But then you can also produce fat on its own. You can produce lipids, and, there's some companies that are using, again, the precision fermentation technologies to produce just molecules of fat, and then those can be, and then that could also be an alternative to, for example, unsustainable plant palm oil production. So
Hmm.
like they're kind of, yeah, a different types of a nutrient, it's a fat produced in microbes.
Yeah. Like making omega-3s, I think one of the Cell fat companies is doing a lot with omega-3s last
Yeah.
time I heard. So yeah, just honing in on those sometimes difficult to get nutrients and making them
Okay.
specifically.
Okay. And this might not be a question that's easy to answer, but is there a difference in kind of cultivating different species? Like
Yeah,
there is.
definitely.
Okay.
Um, yeah, we cover a range of species at our company. And so, for example, like avian cells, so chicken, that sort of thing, you would grow that at a higher temperature. Fish, you grow at a lower temperature. So avian might be like 41 to 42 degrees Celsius, mammalian might be 37, fish might be 27. So when you're thinking about like energy usage and stuff like that, if you don't have to heat them up as much, it's like way more efficient. So I think there's a lot of interesting benefits to the different types of cells. And then, of course, how quickly the cells grow depends a lot on their species as well. You might know more about that, Esther, but I
Yeah,
think it's something to do with how big the animal of origin is or something like that.
yeah, kind of the protein turnover and metabolism sort of differs by animal size. And there's all, yeah, I agree. It's a very interesting topic because, yeah, mammalian cells are generally more complicated to grow. And avian cells, I've heard that chicken cells kind of might be a bit easier, but I don't know whether that's like fully true. It's also tricky because obviously that's not an area where there's been that much research. Like most of we know in cell biology comes from mouse cells and from human cells. Like there hasn't been that much research about even bovine or like fish, there's all sorts of different fish as well. So like it's kind of an exciting area to even see what it looks like because nobody has looked that hard, before. It just wasn't really a question unless you were so super specific, lab. And for example, the other thing is how the tissue is structured. For example, for fish is slightly different. And I've heard that like wild type in the US is a cultivated fish company, and there's a couple of others. And I think the idea is there is it's also a simpler structure to some way. If you imagine like a salmon fillet, like it has these sort of layers that are quite structured, and this is actually easier to replicate than the sort of big interconnected, the kind of slightly messier structure of a steak.
Hmm.
So yeah, there's all sorts of layers to this, but it's an interesting question.
And of course, with trying to make a steak is kind of complicated on much more levels because obviously it's moved, hasn't it? You know, these kind of muscles have had life and and how do you replicate that if you're growing it in a bioreactor?
Yeah, well, one of the things you might be thinking of about what develops the sort of texture and stuff of meat when you talk about the meat having had life at some point is like stretching and moving the muscle. But that is actually something that you see even in the cells on their own. So when you culture cells in a dish, under good conditions, and if they're pretty healthy and stuff, they will spontaneously start twitching and contracting and exercising themselves. Or you can also induce them to do that with like electrical stimuli or sometimes chemical stimuli. Um, but it is interesting that you can sort of get that level of of copying what's actually happening in real life is one of the coolest things that
Hmm.
I've been able to see in the lab. Um, but then if you're looking at more of a macro structure, I think that's where scaffolds would come in a lot, is getting like that correct structure. So sometimes cells, especially muscle cells, when they are forming these fibers, the fibers can be quite random. But if your scaffold is, for example, ridged or something, it can help them align much nicer. And then you might get more of that sort of texture, that sort of thing. And then if you want the full steak with the fat and everything, you might need to co-culture cells. This is a throwback to the very beginning, but when Esther was talking about how Meatable does forward reprogramming, I think what they do is like grow iPSCs, which are induced pluripotent stem cells. They're very early in the lineage, and so they can become almost anything. And so they grow them all together, the ones that are going to be muscle and the ones that are going to be fat. But some of them have been programmed, forward programmed to react to a certain molecule and become muscle, and then the same molecule will make the other ones become fat. And then they're able to like co-culture them somehow. I don't know all the details, of course, but yeah, there's a lot that would go into making a complete textured product. But people have done it. Aleph Farms, I think, or Aleph Farms. I'm not sure how you say it.
Aleph Farms, I think. Yeah. Um, okay. So this is possible now that you can create a full steak experience. I haven't. I mean, I've tried the fish. I haven't tried the meat. And when I tried the white fish, the mouthfeel was. slightly off. It was a little bit sticky, a little bit rubbery, not quite as I know fish to be. So, I mean, how close do you think we are to, to satisfying consumers' uh, kind of meaty desires in a product?
That's a good question. Um, I personally think a lot of the plant-based meats on the market are already doing an incredible job. And so, yeah, I mean, the fact that we're already able to get so close with just plants to me means that like when you're introducing this like last level of refinement, yeah, I mean, I feel like we're pretty close. What do you think, Esther?
Yeah, I mean, it depends because there's so many, like even within meat products, right? You have you have your Wagyu steak and you have your,
Yeah, that's
burger from
true.
McDonald's, which, doesn't have a lot of real meat to begin with, probably. So, like I think it's, we're still a way off from having, full steak at an affordable price. But there's
Mm,
certainly a lot of advancements and a lot of specific applications for, in hybrid products and,
yeah.
these more textured products that, are coming. And yeah, it's exciting to follow along and see how it
So we're not
develops.
so we're not far off, but there are some, there are challenges obviously facing the cultivated meat industry. What are those major challenges?
To me, it's cost. That's always been the big thing. Because yeah, even these wonderful successes of prototype products, the cost is just too much to make it competitive. So that at some point that's like getting the cost of media down, but then when you've done that, it's an engineering problem. So that's
Mm.
what we're working on.
The engineering problem in regard to the bioreactors, building the bioreactors and kind of growing things quickly enough and
Yes, just
yeah,
making the system large scale and efficient. Yes.
okay. And
I'm
apparently,
sure there's many other challenges. I mean, there's
yeah,
biological challenges. There's challenges with everything, but I think when I think about what is keeping it from being commercial at right now, I suppose there's also regulatory. I don't know quite as much about that, but the big thing that stands out to me is cost. But what do you think, Esther?
Yeah, I mean, I think it's a quite a, yeah, clearly the major thing. Um, and it's, I think now we're kind of, I guess that's quite a common view about, there was a sort of hype and maybe 21, 22. There's a lot of companies, and now some of them are going, into administration and going bankrupt. But actually, other companies are sometimes like acquiring IP, for example. Is it Meetable as well? Or I think a company in Netherlands has bought the IP of like Uncommon,
that
who
was
like another
meetable,
company.
Mm,
yes.
So like this consolidation of knowledge hopefully would help to, then, there will be fewer companies, but maybe they will actually achieve this more price parity. And then I think we're not far off from the kind of the better proof of concept, like it's already been available in Singapore for years and it's now available in supermarkets and in some restaurants in the US. But then there's like will be another jump to then hopefully at some point a wider consumer availability, but that will, cost and also just a scale up, just a pure scale of like how much if we want to replace these products, like we just need to produce loads. So yeah.
Mm. So your technology, Alison, in the organization you're working for, is kind of overcoming, or that's the idea, is that they are overcoming the resource limitations for bioreactors. In that perhaps we don't have enough metal in the world, but kind of through creating this extra surface level, uh, then. Uh, extra surface space, you can
mhm.
create more meat in smaller bioreactors, essentially, right?
Yes, that's exactly right.
Nice.
Yeah, perfectly put.
I think I also I the recent news of like this week is has been from an organization called Respect Farms in the Netherlands who've partnered with cultivated meat companies. I think it's Mosa Meat as well, who
Yeah.
who have this project about rather than just going bigger and bigger and having these like absolutely, 1,000 million liter bioreactors. Instead, they want to do small bioreactors, but kind of decentralized production at farms so that also
Oh,
farms
nice.
like meat farmers can convert their eventually, their production systems to work with cultivated meat. And I think it's an amazing project and hope, yeah. they're just starting kind of the new phase of this project where they're partnered with actual cultivated meat companies and the farms
Yeah.
to trial this in existing meat farms,
Well,
but having
well,
these small bioreactors in the farms. So,
yeah, it's brilliant. They've partnered with Aleph Meats
all right.
and Mossa Meat, haven't they, for that? Yeah.
Yeah, yeah, it's
Yeah.
literally this week that the news has broke. So, yeah, it's exciting to see what
It
they
is
will.
very
Wow.
exciting. So
There's
just
this
finally,
wonderful, oh, sorry. Go
oh,
ahead.
sorry, go on, Alison. Go on. What were you
I was
going?
just going to say, there's a wonderful paper called Every Village Its Own Factory. And it was like a speculative paper. I think it's from like 2014 or something. So before many companies even existed, about what cultured meat could be like. And it was very much that sort of vision of like decentralized on farms, all this stuff. And I just love it. I adore that paper. It's wonderful. I wish I could remember the first author's name, but I can't. I can't. But everyone should look it up and read it. It's only a couple pages. It's really good.
Yeah, I think the Respect Farms model is a great one in regard to kind of bringing the public with them because it's a story that people can relate to, and there's kind of real animals living nice lives on farms, and
Yeah.
it can really bring some kind of something that the public can get hold of, a story that they can
Yeah.
follow and in real time, which I think is quite interesting.
Yeah, because that can be a real risk when you introduce lots of technology into the food systems, is that people end up alienated from their food. But with a model like that, people might actually become closer to their food than they are now because it's more personal to go and see like the pig that's donating the cells right next to the brewing reactor that's growing the sausage. And
Yeah,
it's
yeah.
fascinating, it's cool, and the localness, the decentralization, I think is amazing. That's super cool.
It is. It is. Okay, so finally, because we are pushing it for time a little bit, I wanted to get both of you to kind of give an idea of where you think we're going to be in 10 years from now in regards to kind of cultivated meat. What might we be
Hmm.
achieving? Have either of you got any thoughts on that?
In 10 years.
Yeah, that's ages away.
Yeah, I mean, the first cultivated burger, right, from Mark Post was a bit more than 10 years ago now. So, we've come a long way, but there's still a way to go. So I feel like
Yeah.
the hope is that it will become widely available. And like, I think like all of us would have tried a proper culture, like several actual cultivated meat products for sure within the next couple of years. Like people who are within the field, like are, getting there, and then people more outside the field will start to get there as well. I, yeah, it's hard to say whether it would be, so widely adopted or whether it would still be a bit niche, but certainly hybrid products. Like,
Mm.
I don't see a reason why it shouldn't be available because even the regulatory side is like the regulators in the UK for sure are very interested in getting this to the market. And even the EU is slowly getting there, and the US is already there. So
Do you
it's
think
yeah.
do you think we might have on the menu in places like Burger King and McDonald's something which is at least partially kind of cultivated meat within 10 years?
I think it also depends. Sorry, I said I can let you answer this as well, but I think it also depends on the kind of the wider food system, right? Because the problem is also that real meat is actually too cheap. Like it's not. It's getting more expensive because the strain on our food system, the strain on our environment. So it depends whether kind of how the climate change is going to go, whether how food will be recognized or not recognized as a contributor to the wider environmental issues, and food security as well. Like countries like Singapore and Israel were at the forefront of these developments, not really because they were interested in environment or animal welfare, but because they were worried about their food security. So I think it's like a lot of global socioeconomic political aspects to it as well.
Yeah. Okay. Well, let's
Yeah.
well, yeah, okay. So obviously anything can happen in the next 10 years, right?
I think I think I'm a bit, yes, I think I'm slightly more, I think we're such a young industry still. Even though we've been yeah, we're very like she said, the first burger was only just over 10 years ago. And so I would rather we didn't rush and we did it all right the first time, to make sure the technology was really ready before we pushed it everywhere all the time.
Yeah.
And so 10 years, I don't know, I'm just trying to do all this math in my head about it, but maybe yeah, hybrid products and restaurants, but I would be surprised if we were widely um, cost comparable at grocery prices
Mm.
in 10 years. But you know, stranger things have happened.
Yeah. I don't know whether you knew, Alison, the person who is leading the Respect Farms project is the granddaughter of the guy who invented the first burger.
Oh my gosh, Irina?
Yeah, Ira Van Halen, yeah.
Ira,
Ira Van
Ira,
Yeah.
Helen, yeah.
yeah.
Yeah, yeah.
Oh, that makes sense. She's really
Okay.
cool.
Well, it's been wonderful talking to you, and I think we've achieved our objective. Um, so thank you very much for coming on Future Foods interviews and educating us all. And I'll hopefully speak to you both very soon.
Yes, thank you for having us.
Thank
It's a
you,
pleasure.
Alex. Yeah.
You're welcome.